Good morning. Good morning. We have actually a really interesting and exciting panel here. I think we've got some interesting things you're going to hear about here this morning. Let me introduce, let me see what our order is here from left to right. Let me begin with Dr. Axel, who's the Senior Vice President, Therapeutic Head of Oncology and Research and Development, and the Head of Immuno-Oncology at GSK. To my immediate left is Dr. Patrick Soon-Shiong, Chairman and CEO of Chan Soon-Shiong Institute of Molecular Medicine and Chairman and CEO of NantQuest.
Schlom.
Yes, I know. You were missing in action for a minute there. Dr. Jeffrey Schlom, Chief of the Laboratory of Tumor Immunology and Biology at the National Cancer Institute of the U.S. National Institutes of Health. And what do we have over there? There we go. I need to check our order. Dr. Chris Boshoff, Head and Vice President for Early Development, Translational, and Immuno-Oncology at Pfizer. And of course, then Dr. Axel, who's Senior Vice President, Therapeutic Head of Oncology, Research and Development, and Head of, I already did that, didn't I? Grant, I apologize. We're a little bit out of order and I'm a little flustered. Grant McArthur, Professor at the University of Melbourne and the Sir Peter MacCallum Department of Oncology in Australia. Welcome, gentlemen. So let us begin with our order here. I believe, Dr. Schlom, you were going to lead off for us. Each one will have a brief presentation, and then we'll do some question and answer.
And how do we find the slides?
We have, the slides will come up, and there is your,
Don't be scared, I only have 2 slides. I think it's important for us to start off with really defining some of the differences between conventional therapies and immunotherapy, and then I'll discuss combination immunotherapies. And of course, these are generalities. There are exceptions to each of the things I'm going to say. Probably one of the more important differences is that conventional therapies such as hormone therapy, radiation, surgery, chemotherapy target the tumor. Immunotherapy is very, very different because it does not target the tumor, it targets the immune system, which then targets the tumor. The limitations, for the most part, are toxicity in conventional therapies, whereas the major limitations of for immunotherapy is that it requires adequate immune system function. With some exceptions, tumors of large size are not amenable to immunotherapy, and patients who've received multiple rounds of prior therapies and have damaged their immune system may not be amenable. Tumor evolves. There are new mutations, as you've heard about this morning, and there is resistance, which is the major issue in conventional therapies. But the immune system is enormously diverse, as you heard from Dr. Tak-Mak, and there's enormous diversity, and they can target, the immune system, once released, can target multiple thousands of different variants, something called antigen cascade. And the pharmacodynamics are often immediate. The drug goes in, the drug comes out with conventional therapy, or radiation goes in and that's gone. Whereas in terms of immunotherapy, there's a delayed effect and you may need boosters over time. And lastly, with conventional therapy, there's no memory response. Again, the drug goes in, the drug goes out. But with immunotherapy, there is a memory response. We all know we have lifelong immunity to many vaccines, and as you heard about some examples this morning. So, and this is very, very important when it comes to targeted therapy and in terms of immunotherapy and combination therapies. So what are the types of combination therapies? So there are 2 or more types of immunotherapies. Combinations of immune therapies, vaccine, checkpoint inhibitors, that you heard about from Dr. Allison, immune modulators. And there's also the ability of using immunotherapeutics with so-called non-immune-based therapies. That is, immunotherapy, 1 or 2 of them, with conventional therapeutics. So what's the rationale for doing these combination immunotherapies? Well, there are multiple components of the immune system that can be activated by different immunotherapies. We've heard about this morning: T cells, NK cells, dendritic cells, B cells. Relatively low levels of toxicity with most immunotherapies. There are exceptions. And there's a memory response, as I mentioned, with immunotherapy. So one could think of giving a vaccine and then giving a second vaccine or a second agent, such as a checkpoint inhibitor, which can boost that vaccine response. And certain non-immune-based therapeutics can actually alter the tumor microenvironment or the tumor cell and the immunome, that is our peripheral blood, to enhance the therapeutic effect of immunotherapeutics. This is something we've been studying for a long, long time, and it's really a very novel finding, and that is that certain conventional therapeutics that are given will actually alter the tumor cell to make it more susceptible to immune cell killing. And lastly, which is probably the most important point as far as we're concerned, is the availability of these multiple immunotherapeutics. As I mentioned, there are vaccines, checkpoint inhibitors, immunocytokines, NK cells, T cells, immune modulators, and all of these can be used in combination. And that is the challenge on A, is the availability of these agents, and B, how to use them together.
Terrific. Thank you, Dr. Schlom. Chris Boshoff, you're next.
Thank you very much. Thanks for the organizers for giving us the opportunity to be here. So I'm going to build on that. So essentially, what we're trying to do in immunotherapy is take a tumor that's so-called cold, that hasn't got an immune response, and convert that into a tumor with an immune response, but a very specific immune response, because as pointed out earlier, some immune responses are actually good for the tumor. So getting an immune response is actually going to attack the tumor. So that is the challenge. So we believe very firmly that a very broad approach will be needed, will be required to achieve that. And not only developing compounds that's targeting the tumor microenvironment, for example, vaccines, small molecules, large molecules, but also continuing to develop molecules and targeted therapies that specifically targeting the tumor cells. And we know that some of those methodologies can actually enhance immunotherapeutic drugs. So we are developing a platform of different approaches to tackle cancer and to enhance the immune response, including antibody-drug conjugates, bispecifics, vaccines, nanoparticles, CAR T cells, and also other targeted therapies, including oncolytic viruses. And many of these are now in the clinic or will be going in the clinic during the next couple of months. So essentially, our strategy for immunotherapy is to have a backbone, and currently we believe that this backbone will be an anti-PD-L1 or anti-PD-1. So in our case, in Pfizer's case, is avelumab, is an anti-PD-L1, and then we're combining that with various therapies, including with conventional chemotherapy, with other immunotherapeutics. We're combining with antibody-drug conjugates, as well as with targeted therapies. So, for example, tyrosine kinase inhibitors. Like all of you, we wish there will be one day a blood test, that we can take a blood test and then predict who will respond to immunotherapy or who will be resistant, or what will be the prognosis for that specific individuals. So developing biomarkers. This is becoming more and more challenging, especially if we're starting to combine therapies. With combination therapies, biomarkers are complex. We're also starting to explore the so-called microbiome, those bacteria in the gut. Essentially, we know now that potentially some of these bacteria can help immune responses, and potentially in the future, some of these bacteria could be used as a therapeutic to enhance immune responses as well, so-called microbiomics. So to do this, we're developing basket studies, and actually one of our biggest basket studies now is our phase 1 study with avelumab. Has recruited over 1,600 patients. So these are big, big basket studies, and we also using these basket studies for combinations with antagonists, with agonists, or with targeted therapies. And then, of course, we're starting to explore mechanisms of resistance. We know that the majority of patients will not respond to single-agent checkpoint inhibitors, and most patients responding will actually progress on checkpoint inhibitors. So understanding why patients are inherently resistant, why they do not respond, or why patients have adaptive resistance, why they become resistant to therapy. And then my last complex slide is actually an illustration of our current combinations that's in the clinic or combinations that's planned to be in the clinic during the next couple of months. So as you can see, avelumab is developing as a backbone, as an anchor for many of these IO combinations we are developing. And then, of course, we're also combining with targeted therapies and with chemotherapies, and perhaps most importantly, many of these are now starting to be with collaborations with academic centers like MD Anderson or Yale, and also collaborations with the NCI and with other industry and biotech companies.
Thank you. Dr. Patrick Soon-Shiong.
Thank you very much. It's a pleasure being here and an honor to be here. You know, we've taken a very different approach. And the approach is a philosophical one, and maybe a very complex scientific one, where we believe that really cancer is a normal physiological event of regeneration. That cancer is merely a misread as stem cells grow. And in fact, as we're sitting here today, each of us are generating cancer cells, but we've been born with the ability to protect ourselves through the thing called a natural killer cell. And as the cancer cells get smarter, it actually finds ways through generations of its mutations to put the T cells and dendritic cells and NK cells and B cells to sleep. So the key is to really understand what is these, what we call abnormal epitopes or mutations that are specific only to your tumor, because you are your tumor, meaning you are your genome, and the genome generates the tumor, which then leads to a challenge to actually generating a whole genome sequencing and the proteomics to identify what we call the neoepitope. If we can do that, we can then eventually, and eventually is actually now, generate a vaccine which we can engineer to give you multiple occasions to educate, re-educate your body so that it actually can activate what's in your body to go kill the cell. We call that the cancer vaccine, and for the rest of my time, if I could just show the video, and I'll be speaking more about that this afternoon.
Every year, over 14 million people worldwide are newly diagnosed with cancer. That's why we created Cancer Moonshot 2020 with the ambitious goal of changing the course of this deadly disease by developing a cancer vaccine by the start of the next decade for all cancer types at every stage. To reach that goal, we must learn how cancer hides from our immune system, because each of us are already equipped with the right tools, our own immune system, to kill cancer cells being formed in us every day. To accomplish Cancer Moonshot 2020's goal, we're accelerating trials of new treatments based on advances in technology that once seemed impossible. To understand the path to a cancer vaccine, first, let's take a look at how our immune system works. Central to it are natural killer cells and killer T cells. They roam around the body, searching for cells that are infected or damaged, like cancer cells, and are able to seek them out and destroy them. The conductor of our immune system is the dendritic cell. By absorbing and processing the material it recognizes as foreign, in this case, the abnormal peptide sequence inside the cancer cell, it begins to understand which cells to target. The dendritic cell, now activated, is then able to educate the killer T cells and natural killer cells to wipe out cancer cells. But cancer is smart. Over time, it figures out a way to turn off our immune system by putting natural killer and T cells to sleep. It then grows and continues to mutate, evading our own natural defenses. So how do we turn our immune system back on to fight against these clever cancer cells that have hijacked our cells' natural biology? We start by taking a sample of the tumor and perform a test we call GPS cancer to uncover these mutations and protein sequences. Then, the GPS cancer supercomputer rapidly identifies and decodes the abnormal mutations and proteins produced by the cancer cell, what are called tumor-associated antigens and neoepitopes. We are then able to manufacture a cancer vaccine, customized to your tumor and your genome, by loading that discovered sequence into a specially engineered common cold virus called an adenovirus. The cancer vaccine with the neoepitope is then injected under the skin like a flu shot, where it arms the dendritic cells with the tumor's hidden code. The activated dendritic cells then train the killer T cells and natural killer cells to seek out and destroy the cancer cells that contain this specific amino acid sequence. This uniquely engineered vaccine is also redosable, meaning it can be given multiple times, like a booster shot, to help build up your immunity. With this vaccine tailored to your specific cancer, the cancer is no longer able to hide from your immune system and will put us on the path to orchestrate our immune system to win the war on cancer by the year 2020.
Interesting, and I'm sure we have many questions surrounding that. Let me pass our slide
Thank you.
down again. And Dr. Hughes?
Good morning. It's an honor to be here. What I'd like to do is give you a brief holistic view of where the field of cancer immunotherapy, which we now actually call immuno-oncology in industry terms, might be heading. In order to understand where the future might go, it's useful to have a brief understanding of what the past has brought us. Let's start with a quick look at what has the field delivered so far. You know the history of cancer immunotherapy is actually long. There's more than 100 years of an attempt to use the immune system to treat malignant disease. We turned a corner on this really just in 2011. And since 2011, we have seen 3 types of generations of immunotherapies. The first one were the initiating agents, and you heard from the immunotherapy panel earlier, checkpoint modulators were at the forefront of this. T-cell therapies came next. So you see here, generation 1, Yervoy was the first product that is now being widely used, and it has already been expanded on with generation 2 with the PD-1 blockers, as you have also heard about, and a series of new therapies that are pushing into the space and expanding our toolbox. So we have many more tools to work with. And it introduces complexity that is probably best addressed through combination therapy and through the underlying science that teaches us what to combine. So let's have a look at that. This is the trends that currently exist in the field. We have started replacing standards of care, meaning in most cases chemotherapies, through life-saving immunotherapies. You saw that in melanoma, beginning to see it in lung cancer, and it happens in other diseases. That comes with substantial improvements of survival, something that most conventional therapies have unfortunately not been able to deliver. Now, a next frontier that we are working on very hard, and you have heard that already in some of the previous presentations, is immune profiling, understanding the immune status of a patient before you treat. And then, of course, how it changes with therapy. That would enable us to identify who should get what therapy, or in an extended way, what combination. Complex combinations are the consequence of this. Transformational effects, which means maximum benefit for patients, cannot come from a single agent. In all likelihood, it will come from combination therapies tailored to the individual patient's immune status. So we're working also on improving endpoints to be able to measure that more effectively, because immunotherapy does not behave like chemotherapy in a patient, and you don't always measure the same things in order to be able to declare an effect. And then finally, we're expanding our toolbox. And that's where combinations become particularly relevant. The tools you use to put together will enable ultimately the effects you can achieve. So if I look at what does the toolbox look like, so this is the spectrum of modalities that we use in immuno-oncology. If you read this slide from top to bottom, the blue boxes show you the branches of the immune system. In a very simplified way, adaptive immunity is the cells that can respond very specifically to a certain threat, and innate immunity are the fast responders that are less specific. So in the adaptive immunity space, you have the T cells we heard so much about, and you have B cells that make antibodies. Everything underneath those blue boxes are different types of treatments you can use to engage the immune system and make it fight cancer. So you can start with cytokines. These are signaling molecules. You can go to cell therapies, which are the cells themselves that we manipulate and give back to the patient. We can go to vaccines, where you provide an immune stimulus to generate an immune response. You go to T-cell checkpoint modulators or other checkpoint modulators that affect other parts of the immune system. Those are the molecules we heard so much about, CTLA-4 or PD-1. They are connecting bispecific antibodies. There are other bispecific antibodies that enable you to engage two targets at the same time in one molecule. They are small molecules. All the targeted therapies that you heard of are often small molecules. We are just beginning to really use the potential of small molecules in immuno-oncology, mostly to change the environment of the tumor so T cells can be more active there. We have oncolytic viruses, so basically we construct a virus that infects tumor cells, makes them burst and release antigens, so you can actually induce an immune response that can then be modulated with other immunotherapies. And finally, you have adjuvants. The concept of adjuvants has been used in vaccines, prophylactic vaccines for infectious diseases very commonly. We're now carrying that forward into therapeutic approaches against cancer. So look at this universe. It's a very big spectrum of opportunities here, different tools in the toolbox that we now need to scientifically combine. And that is our challenge. And I'll say one more thing about this before we come to discussion. So the aim obviously is we want to cure patients. And we can use the cure word, which has been hesitantly used in the history of oncology for so many years. We can use that more confidently today. And in order to achieve it, what do we need to do? The first is triggering the right immune response. And you need the right tools in order to do that. Beyond triggering the immune response, you then want to be able to modulate that immune response, and our checkpoint modulating antibodies may be the right tools to do so. Further, you want to induce a danger signal. A danger signal basically means you're creating a signal that brings the immune system towards the cancer. You're sculpting the tumor microenvironment. You're basically preparing the tumor to receive T cells so they can do their job. You do specific tumor targeting. Some immunotherapies are actually very specific against only one tumor target. The CAR T cells we heard about belong in that category. You look at memory, so the effects you induce are actually durable over time. The immune system remembers and can actually produce an effect that it goes way beyond that of a chemotherapy or targeted therapy. And finally, you want to maximize the effector cells, which are the cells that do produce the effect. Those are often engineered. So T cell therapies can be particularly potent effector cells. So if I look at that in totality, there are three big C's in the space of immuno-oncology. The first is our trend towards combinations. The second is that of collaboration, because no single player, be that in academia or in industry, can do that alone. And the third is then the cure word. We actually have the potential here to achieve transformational effects and cure patients. It's the first time in the history of cancer that we can say that with confidence. And I think we need to take that opportunity.
Very good. Thank you, Dr. Hughes. Take that back. We'll pass that down. Last but not least, Dr. McArthur, you're up.
Thanks very much, Max. And, you know, isn't that exciting to hear the word cure mentioned? And that's absolutely what we're all aiming for. But I'm going to bring us down to ground a little bit, you know, back to reality, because we do have some challenges before we're getting a lot more patients cured with cancer. And basically, we need to continue to innovate if we're going to cure more patients. So I'm going to tell you about a new way of doing clinical trials, because clinical trials are slow, they're expensive, and they're actually prone to failure. They cost a lot of money, and sometimes they don't show us a positive result. And we also have to be very cognizant. We're spending a lot of money, a lot of effort investing in new therapies, but we have to make these therapies available worldwide to all patients with cancer, including those in developing and low-income economies. And this is one of our challenges. There are answers, though. So what about clinical trials? So we've heard a lot about clinical trials at this meeting. So the traditional way we do a new trial, after we work out what the dose is and how to give a drug in combination, is a phase 2 trial. Now, I'm going to use the example that actually came up from Jim Allison this morning. and that is adding immune treatments to targeted treatments. This is something in the melanoma field we're very passionate about. Jim talked about it. We want to kill the cancer cells by targeting the oncogene, the BRAF, that keeps the cancer cells growing, but then activate the immune system at the same time. So one hypothesis is we use the tablet drugs to target BRAF, and we use the PD-1, this remarkable new therapy, to turn on the immune system. So traditionally we would treat 50 to 100 patients with a new treatment, measure how many responded, how long it took for the cancers to grow again, if they do grow again, and then have a guess that, yes, that looks pretty good, let's go to phase 3. So then you put hundreds of patients on the old treatment, on the new treatment, 50-50, and see which the winner is. 60 to 70% of the time you get it wrong, and it's negative. And I put it to you that that's not good enough, especially for patients that need new treatments quickly. So what we're doing to innovate, given this enormous challenge that we've heard about all these combinations. Just in melanoma, the disease that Jed Walchok and I treat, you know, there's over 1,000 different possible combinations. So we have to innovate in that space. We also need to be really sure early on that something's a winner, so that we can invest, most important of all, patients that are participating in clinical trials, that they're investing their time and effort in being enrolled in a clinical trial that has a high probability of success. So how do we do that? So what we do is we call it an adaptive platform trial. So here we don't just evaluate one new therapy, we evaluate multiple new therapies. Initially, evenly, we look at the biomarkers, and the trial that we're starting, we're looking at the BRAF biomarker, we're looking at biomarkers called PD-L1 that can help predict whether or not the immune system is going to work. And we're really delighted to be collaborating with Patrick in this space because of the technology that he brings about tumour profiling. So initially, patients get allocated to multiple arms evenly, but then adaptation occurs. It's not just a toss of a coin, it's sort of like a clever toss of a coin that starts to favour the best treatments. So more patients get enrolled on the treatments that are doing best, and eventually you get to what we call graduation. And that is that the best treatments then go on to the phase 3 with a high probability of success. And of course, some treatments won't work, and so they drop out. And we see this platform happening. We're doing melanoma because we see cure in our sights in melanoma, but this can be applied to any cancer. So we do have some challenges, but I put it to you that we can overcome them. Now, the second big challenge is the cost, and I'll just give you an example here in my field of melanoma. So these are the treatments we heard about this morning, the pioneering treatments, remarkable results, and the cost. You're talking $150,000 plus in US for these treatments. You start adding them together, it goes up even more. If you do adjuvant treatment, the way it's approved, the CTLA-4 treatment, we heard about one of those break treatments this morning, it's over $1 million for a course of therapy. These are extremely expensive. How are we going to get these treatments to patients worldwide? And Max, I know this is something that we can discuss and it's come up earlier in the meeting, but to me that's one of our challenges, that together with STEM for Life, with the Pontifical Council for Culture and everyone in this room, we can solve that problem. Thank you.
I hope so. I hope so. Thank you. So as we get started here on our discussion, some of these questions will be, I think, more specifically aimed at individuals, but I think some of them are questions that we can all weigh in on, and I know that our panelists have somewhat different points of view and opinions on these, so I ask you to, it's okay to chime in and give me your opinion on a particular take here. But just so we're clear, by the way, especially because we have so many lay people in the audience, some of the rationale behind combination therapy is that we have had some monotherapies that have had some success, but I guess the metaphor is no fortress, in this case a cancer cell, is likely to be overcome or overrun by attacking it at a single point. You often need to attack it from multiple points in order to destroy it, and cancer cells, as we've already heard, have multiple ways of escaping the attack. And so by coming at it in a variety of different ways, 2, 3, or more different ways, we may be able to finally overcome cancer. I call it death by 1,000 paper cuts. Is what I'm hoping we can achieve here. And it was pointed out to me earlier by this morning by Dr. Hus that we have in fact been doing combination therapies for a long time, and both in chemotherapy and combining chemotherapy with radiation, surgery with chemotherapy and radiation and so forth, but it's been somewhat random up to this point. We are now trying to move into an area where we decide these combinations in a rational manner. So, Dr. Boshoff, how do we decide what is a rational combination? As Dr. MacArthur said, there can be thousands and thousands of potential combinations. We can't test them all.
So we are using, all of us are using preclinical models, but we know that many of these preclinical models have limitations, and particular limitations dealing with the immune response, which is very different from preclinical models than it is from humans. So some of it, although some of our data and some of the studies we will be pursuing will be based on preclinical models, some of it will be based on biology, where there's biological sense. So, for example, we combine, as mentioned earlier, combining a targeted therapy that kills tumor cells, then releasing antigens with a checkpoint inhibitor that can then release the T cells to attack the cells. So that's a rational combination. So although it may not work in a preclinical model, that we could argue is worthwhile testing in a clinical setting, in a clinical trial.
You said you looked like you had something to say.
Yes, I do have something to say. And some of this, I totally agree. There's a lot of work in preclinical models that informs us. But
Tell me, just to be clear, again, so everyone understands, what is a preclinical model?
Preclinical model, there are basically two major types of preclinical models. The first is we use a mouse that has a tumor, a spontaneously arising tumor or an injected tumor, and we give it various therapeutics to see what cures the mouse. A lot of things work in a mouse, that's the problem. We then use human cells, human dendritic cells, human T cells, and activate those human T cells and see if those, but in different ways, and see, do those T cells or NK cells kill tumor cells more efficiently? Which combinations really rev up a T cell or rev up an NK cell to make it a much better killer of a human tumor cell? So we do that. And we also use so-called athymic mice that don't have an immune system, so we can put human tumors in a mouse, and then we can inject different therapeutics, different ways to see how those human tumors are destroyed. One of the major issues that is to answer the question, one of the major issues has been the ability of immunotherapy to target the big tumors, the big breast cancer, colon cancer, prostate cancer.
Especially the solid tumors.
The big solid tumors, the big killers that kill 85%. Now, it's been very, very effective, as you heard this morning, in lymphomas, leukemias, lymphomas, and melanoma. And there's been immunotherapies in melanoma, for a while, but it's been amazing how well it's worked there. One of the issues that's come up is there's something we term, different people have different terms for it, that melanomas are, quote, inflamed tumors. They have T cells in those tumors. The body is normally attacking those, attacking the tumor. And the brilliant work of Dr. Allison with this checkpoint, he just released the brakes. The tumor builds up a defense mechanism when it sees a T cell. And this drug that was developed will just releases that brake and lets the T cell do its dirty deed, killing the tumor. One of the things we're finding, and people know, is that tumors like colorectal cancer and pancreatic cancer and breast cancer are not so-called inflamed. They don't have a lot of T cells in them to begin with, so that these checkpoints really don't work very well because there's nothing to release the brakes on. Enter a vaccine. So people like me and a lot of other people have been using vaccines for a while. And what we're finding is, yes, they work in some patients, and we've got a fully accrued phase 3 study in prostate cancer now. But one of the issues is, as the T cells get to the tumor from the vaccine, the defense mechanism comes up. So therefore, you want to release the brakes, use these checkpoint inhibitors.
There's your combination.
There's your combination. And indeed, We've published recently, I think the only trial published, where we've used ipilimumab, this checkpoint inhibitor, with a prostate cancer vaccine with some very promising results. And we have many, many trials on the way, some ongoing at the clinical center and the NIH with our collaborators, with all these various kinds of combinations. I think this is what's going to be very exciting in the future.
Now, Dr. Soon Chong, I suspect you have a slightly different take on this. Would it be fair to say that this vaccine that you described in your animation, in your presentation, in a sense, does the combination for you?
And I wouldn't describe it as a different take. I think I agree completely with what Jeffrey says. It's maybe a little more nuanced in the sense that we are trying to activate, actually, what currently exists in your human body. So the way to think about it, and one of the slides that both of them showed, is where we were trying to get to is the memory cell. And when you see memory cell, and you get to the cure. But before you get to the memory cell, you actually have to get into a thing called the tumor microenvironment. You actually have to get into the tumor. And the way I think about it is hand-to-hand combat, as opposed to a war, a battle. And the hand-to-hand combat really involves all of these cells simultaneously. The NK cell, the dendritic cell, the T cell, the B cell. So it's a very fine orchestration of these cells simultaneously once you get into the tumor microenvironment. I think it wasn't very well appreciated, the purpose of our invention.
You'll block yourself out.
Sorry, okay. Purpose of our invention of Abraxane was to develop a nanoparticle of albumin to get into the tumor microenvironment and perturb and create what he calls a danger signal. Once you have the danger signal, your body actually is tuned with the NK cell to find any cells that have a danger signal. You activate the NK cells, but your body now has not been accommodated to what is these abnormal sequence. And we solve that, not through biomarkers, but actually creating what we call liquid tissue by taking paraffin blocks of the patient's own cancer and doing this whole genomic sequence and proteomic analysis. I think what is scary and concerning is that we've now discovered a thing called heterogeneity, meaning in the same patient on the same day, from the same biopsy, but from the same tissue, there might be multiple cells. So it's a multiple clonal disease. So we need to accommodate for heterogeneity, and the only way you can do that, actually, instead of killing the cell from inside out, you have to kill it with targeted therapies. You need to kill it from the outside in with cytokines and with granzymes, with the natural killer cells, with the killer T cells. So it is a very delicate symphony. And that symphony and that combination of symphonies is now what's exciting. We can, through quantitative analytics and proteomics, know in real time, through minimally invasive biopsies, what's going on. And unfortunately, we have to not only track it on every patient, but modulate it based on every patient, treat it, and I think we can get to, in fact, I know we will share some of the data today at my talk, get to the memory cell and have patients alive 5 years, 8 years, 10 years in what we think of HIV today or chronic hepatitis, where they're not cured of the disease, but they're living with the disease with a high quality of life. And I think that's where we're actually very much on the precipice of doing that.
But now these tumors that are heterogeneous at the point that you have started, or you're starting to develop the vaccine, do you need to then remake the vaccine because the tumor may continue to mutate and accumulate different clones?
Yeah, that's a good question. First of all, because of the heterogeneity, this is where the old chemotherapy that we actually use actually is useful, because you can now indiscriminately use chemical poisons, but not to poison the entire body, but actually to stress the cell. That's why I believe in what I call low-dose metronomic chemotherapy. Secondly, when we do genomic sequencing, you have to do what we call the whole genome, 3 billion base pairs, 300,000 times 2 against the tumor normal, 200,000 RNA, 10 million proteins to find these mutations, and there's thousands of them. But the mutations that are important are not the mutations that exist. The mutations that are important, the mutations that can be recognized by the dendritic cell called presented. And we can now isolate those, and if they're down to 50 to 70, excitingly, we can actually put all of them into the adenovirus. Now you've actually addressed heterogeneity, you've addressed the concert or the symphony, and again, we'll show you some of that, and that's what the moonshot is.
Very good. So, Dr. Hoos, we're talking about a lot of these combination therapies, but there have been some, for lack of a better term, roadblocks to actually testing these. What are the challenges here to testing some of these combination therapies, and how do we overcome that?
Yeah, so the simple challenges are defined by the rules of drug development. Not all drugs are testable in the stages of disease where you would like to test them. It goes through stages. So for example, if you have a new drug and you haven't proven its effect yet, it's often being tested in patients with very late stage disease. And as it then shows effects, it gets tested in earlier stages of disease. It's a slow process. It doesn't enable us to really move fast. So we're revising that paradigm, but it isn't trivial. And if you introduce new combinations, it is a similar kind of situation. So the so-called, life cycle management of a drug includes the testing of it in increments in new settings. So we need to improve on that, and we need to get a bit more courageous. That means don't get hung up with incremental steps. Aim for larger improvements, transformational effects, as we call them now. And you can achieve that with combinations. But it does take much more courage and investment. So having said that, there are, of course, other factors that matter. Collaboration, as I said it earlier, is at the forefront of what this field needs to do. And actually, since the beginning of immuno-oncology, for the last 5 years, we have seen a substantial increase of collaboration in the field between academic centers, between industry partners, and of course between academic centers and industry. And it's staggering how much more collaboration there has been. Some companies have one attractive molecule that combines well. They may combine it with 30, 40 different partner molecules owned by other companies. Of course, there are nuances to how these collaborations work. Who owns what at the end of the story, who pays for what, these are things that need to be decided. But actually, I'm very encouraged by the direction that it's taking. We're doing much more now than we ever have in the history of oncology. So it's a good thing for the most part. And we need to continue to encourage this. And that's why I'm encouraged by things like the start of the Parker Institute, which basically gives us another mechanism to provide access to combinations. And as you can see, funding for immuno-oncology from the industry side is significant. There's a lot of resources made available to understand the immune system, to find new targets, and to then put new molecules into clinical testing. But no company and no academic center and no nonprofit organization can do it alone. It has to be a collaboration. So the big C for collaboration is really important to solve the problem.
And the other big C, though, here, there's the cost of developing these therapies, but then there's also the cost of delivering them. And how are we going to make these affordable? How about you take that?
Yeah,
Dr.
I think
McArthur,
this
'cause you mentioned
is
that.
a big one.
What do we do?
Yeah, and I think we need to think globally. Okay, so not just in higher income countries, we've got to think about lower income countries. So, I think we need to learn from history. And we had an enormous public health challenge in the disease of HIV. It's not finished yet, but we've had enormous advances, particularly in combination drug therapy. Interestingly, the same thing this whole session is about for cancer. And the combination drug therapy for HIV has now turned HIV into a chronic disease, and there's access to drugs in the developing world. So I think we should learn from history. So how did it get to the point where patients in low-income countries could get access to HIV drugs? Well, it was really a combination of high-income countries' philanthropy with a lot of government money and non-government funding sources getting together to help source education of healthcare workers, but also access to drugs. And also the pharmaceutical industry recognising that countries with low ability to pay, they get drugs at lower cost. This is a very important point, at lower cost. And that, for HIV, included letting generic drugs being produced in countries like India so that at lower cost, so that Africa and other places in the world, in Asia with high rates of HIV, could get access to drugs. So I would put it, to us as a group here in this room, that let's learn from HIV. We're at an incredible point of time for cancer. We're actually getting some wonderful drugs that are leading to such miraculous effects in people's lives. We've heard several stories at this conference, so let's learn from the past and consider some of those lessons to get access for patients.
So Dr. Soon-Shiong, is there a way to deliver these things in an affordable manner?
Not only I think there is a way, I think that's both a moral and a financial imperative. I think it's impossible. I think if you see this $1 million drug. But what's exciting, again, I have maybe a slightly different nuanced view, is that I believe that actually the biological pharmaceutical factory of the future is actually your human body. So in so doing, by actually inducing this vaccine, and we're working with Sanford Health, and I think they're here, where we're actually doing HPV. We've created our vaccine with HPV, and cervical cancers, is one of the, in the developing countries, head and neck cancer. So the ability to actually give a flu shot together with these other combinations actually subcutaneously, which means you can actually literally give it in any community. One of the things we're doing is working with community oncologists in the country. We're even working all as far as the Navajo Nation. I think it's an imperative as part of the development protocols, you think this through as a global issue for humanity as opposed to an American issue. And I'm encouraged by the fact that I think we're going to be working with organizations like Sanford and go to places like Africa and India. In fact, we'll be announcing something in India soon.
Good. Well, obviously, we need to figure out a way to deliver it globally. Dr. Boshoff, tell me, everything we've heard this morning has pointed us towards the future of cancer therapy is immunotherapy, to the point where we've almost forgotten about everything else. Is that the future, and is that where we should focus all of our energies, or are there still some other areas that are worth exploring?
So we believe definitely that a very broad approach is needed. So for melanoma, that is clear. There's less and less patients with melanoma that will now receive chemotherapy. Beyond melanoma, it's much more challenging. If we look at the data for second-line lung cancer, for example, with a checkpoint inhibitor, 30% of patients will be alive for 24 months, which means 70% of patients will be dead at 2 years. So there's a lot of room for improvement. So we believe to really enhance immunotherapy is not only about combining immunotherapies, for example, a checkpoint inhibitor with an agonist, with something that stimulates the immune system, but to combine also with conventional therapies. And we have a number of studies currently with chemotherapy and checkpoint inhibitors. For that specific reason that Dr. Schlom mentioned, is with immunotherapy, you can potentially get this inflammation within the tumor, and then you come in with your immunotherapy. And also other approaches. We should continue to really develop drugs that actually target the oncogenic pathways, those pathways which the tumors are addicted to. We know tumors become resistant to those, but if you use some of those in combinations with chemotherapy or with targeted therapy or with other targeted therapies or with immunotherapy, then I think we're really going to start to transform this disease. So I firmly believe that we can't just focus on immunotherapy. Immunotherapy will become one of the anchors of future cancer therapy, but other treatments like targeted immunotherapies or targeted cytotoxics, small molecules, targeted therapies, TKIs, they will continue to be anchors of therapy in the future.
Other combinations, right?
Other combinations, not only IO.
So Dr. Schlom, kind of to finish up here, because we have, as you may know, a fairly hard out. We have a couple of important guests downstairs.
Would that be?
It's a surprise. The best, the worst kept secret here. I think it's something that has been that is now pretty common knowledge amongst oncologists and scientists, but less so, I think also amongst the lay public, is that what is important now and will be grow to be more important in the future is cancers will need to be very specifically identified, not by the neighborhood they happen to be growing in, not by the organ they're growing in, but by their specific genetic and biochemical markers. How important is that piece of it in deciding what are important combinations or even what therapies to begin with?
Yeah, that is where the world of genomics and immunotherapy come together. And Dr. Sun-Chang talked about being able to, I don't know if it was appreciated from the slide, from the presentation, but there is now a CLIA certified test where on from a paraffin block, you can put a slice on a slide, and this can be the DNA mutations, the RNA mutations, and mutations that go into the protein, which is what the immune system sees, can all be identified. Now, this is not a dream. This is now CLIA certified.
And these are not live cells, clearly. They've
Right.
been fixed
These
and
are
put
from
in
your
the
blood,
paraffin.
from the body. I mean, it's hard to believe, but it's true, all right? And so this is where you can identify those specific mutations. Now, the other thing I just flashed up the enormous diversity of the immune system is one thing. You know, when you take a vaccine or a targeted therapy or whatever, and you target a particular mutation let's say with a vaccine, what we have shown in preclinical models, and we have evidence in the clinic, although we can't do these things in clinical studies, is that many times it's not the antigen that's in the vaccine that you're given that kills the tumor, but it's when the tumor is destroyed a little bit, it exposes all its mutations and its antigens to your immune system, and they then cascade in and then cure the tumor. So the patients who have done best are the patients that show this cascade effect to a vaccine that is not just to the antigen that's in the vaccine, but to other tumor antigens. So it's just that there's an enormous diversity of the immune system that is not quite appreciated. And I think this is going to be something that we can really take advantage of in the future.
I call it the cancer cell has donned the Harry Potter cloak of invisibility.
Yes.
You start to take a little bit of it off, and suddenly it's exposed, and the rest of the immune system can go after it.
Yes.
I had a daughter who read a lot of those books. That was part of it. But let me go back just to finish up, just to make sure that we're clear. So what we need to do is, whether it's a prostate, colon, breast tumor, characterize that tumor. And that is what, and that's now possible, is what you're saying.
Yes, and actually, this is actually in practice now. This is what we're doing with Dr. Sun-Chang. We're starting off giving the patient a quote, conventional vaccine to tumor antigens that are shared among prostate cancers, okay? So they get that. It takes about 4 months, 3 to 4 or 5 months, to make this patient-specific vaccine, and then we're going to boost with that patient-specific vaccine. So, you know, it's just like a 1-2 punch. And also, you know, trials are underway where we're using vaccine, combination checkpoint inhibitors, immunocytokines. All of these are actually in the clinic now, and I think this is going to be a very, very exciting time.
What I'm told is that some prostate cancers may actually be more similar to breast cancers than to other prostate cancers. Is that fair?
Well, I think what's going to be very exciting, as Dr. Schlom has mentioned, is we're actually at the verge now where you'll have these preventative vaccines where, you know, early stage prostate cancer, Gleason 6, which is just active surveillance. You'll have an injection of a prostate cancer vaccine, like Gardasil for cervical cancer. And that is very, very, very close and very real.
Just add one thing that Dr. Sun-Chang said in terms of surveillance. There is a vaccine trial ongoing actively in many centers, in prostate cancer, active surveillance versus vaccination. These are patients with men with low-grade prostate cancer, Gleason 6 or below, and the doctor says, well, you know, we can give you all kinds of therapies, but just wait and see. And so this is really the beginning. This is active surveillance in one arm, this is a randomized trial, versus vaccine in the other. And these patients are going to be biopsied after a year and see how well they're doing. So this is ongoing in multiple centers right now.
So I want to thank our panelists, and I think if there's a take-home lesson that I'm hearing both yesterday and in the panel so far today, is that collaboration here is what is going to solve the puzzle. Collaboration between scientists, also between different arms of industry. We need to break these silos down. That's what we've heard Sean Parker is busy doing, but there are a number of other initiatives trying to do essentially the same thing. So don't keep your data to yourself. Share it with somebody else, please, because that's what's going to make a difference. Now, before you start, I see some people bolting already on me. We're going to go downstairs to the Città del Vaticano Sala Nervi. How did I do? Was that good? Oh, good. Where you can join your families and our other guests there for the keynote address by the Vice President of the United States, Joseph Biden, for those of you who haven't been following politics. And that will be followed by our audience with His Holiness Pope Francis. Today we will also, we have a special treat, be honoring our pontifical heroes. And we understand that the Holy Father will have a special surprise for our heroes, so you won't want to miss that. And then we'll tell you a little bit more about them individually when we come back upstairs. So head down. Remember, no photography down there. Behave yourselves. That's all right.
Thank you, this is wonderful.
Thank you. How do we do? We covered what we...
Thank you. Very much.